Okay, so this is the first recording of the reproductive system. So the reproductive system is characteristic of living organisms in general, and essential for the survival of the entire species. It is not technically necessarily required for the survival of the individual. This system is in a state of hibernation in humans until puberty, period in childhood, when reproductive organs mature and become functional. Now, there are similarities between male and female reproductive organs. The gonads, which are the primary sex organs, this is the most important organs for both genders, where secretion of sex hormones, including testosterone and estrogen, will occur. The gonads will also produce the gametes, which are your sex cells, sperm and egg. Both genders have additional organs and structures called accessory reproductive organs that will contribute to the functioning of their respective systems. Cell division can occur either by mitosis, which is normal cell division, in which you start off with a single cell and divide into two daughter cells, and that will produce identical daughter cells, or by meiosis, which is a special form of cell division that occurs in cells that are destined to become gametes. The difference between mitosis and meiosis is that mitosis will be a diploid cell, result in diploid cells. Meiosis will result in haploid cells. We'll define those in a second. So fertilization is a process in which a sperm and egg cell fuse to form a new single-cell offspring, which is referred to as a zygote. The zygote eventually divides to produce all cells of the new individual. They must contain the correct number of chromosomes, half from the ovum, half from the sperm. And in this case, for humans, it is 23 pairs of chromosomes. Again, 23 chromosomes from sperm, 23 chromosomes from the ovum. Meiosis is the process in which cell divides to form a daughter cell with half the number of chromosomes, ensuring the correct number in gametes and eventually the correct number in the zygotes. All human somatic cells have 46 chromosomes, or 23 pairs. And because they're 23 pairs, we refer to those cells as diploid, because they have a full paired set of chromosomes. Paternal chromosomes, these are from the individual's father, and maternal chromosomes are from the mother. Each member of a pair of chromosomes has the same genes, segments of DNA that code for specific proteins. Pairs are called homologous chromosomes. Matching genes on homologous chromosomes may have different variants or alleles. When a cell is not undergoing division, its DNA is in the form of chromatin, long pieces of DNA and their associated proteins. After replication and as the cell division begins, chromatin threads coil tightly and condense into bar-like chromosomes. Now, before meiosis begins, the cell's DNA is replicated so that each new daughter cell will get a complete identical copy of the chromosomes, or sister chromatids. Meiosis proceeds through 4 basic phases: prophase, metaphase, anaphase, and telophase. Phases occur in 2 successful divisions, unlike mitosis, where meiosis 1 is the first meiotic division and meiosis 2 is the second. There is often confusion as to why daughter cells at the end of meiosis 1 are haploid, because they might appear to have the same absolute number of chromosomes as the starting mother cell. However, remember that you are not seeing 4 individual chromosomes. You are seeing 2 sets of sister chromatids, simply 2 identical copies of a single homologous chromosome. Mitosis occurs because new cells are needed for tissue growth and repair. New cells must be genetically identical to the original. Mitosis occurs by first making a complete copy of the DNA and then dividing the cells so that each cell gets 1 copy of that DNA. Meiosis, on the other hand, produces sperm and ova for reproduction. Cells need to have half the number of chromosome of the original cell, so that when they combine to form a new cell, the chromosome number will not be doubled. So you're essentially going from that diploid to that haploid there. Also, meiosis mixes up the possible combination of genes that each sperm and ovum will receive, producing genetically unique individuals. Unique set of steps that reduces chromosome number and shuffles the alleles, referred to as crossing over, is referred to as meiosis 1. The completion of the process to potentially form 4 genetically unique daughter cells is the process referred to as meiosis 2. Here's a comparison between mitosis and meiosis. Just again, look at the different sister chromatids there so that you are aware of what's actually happening here and the difference between the diploid cell and the haploid cells. Okay, so now we're going to explore the male reproductive system. The male reproductive system consists of the sperm-producing paired testes, which are the male gonads, within the scrotum. Sperm travels through the ducts that leads to the opening of the penis. Accessory glands will secrete semen and the fluid that accompanies the sperm through these tubules or ducts. So here's the internal organs in the male reproductive system, looking at a mid-sagittal cut, and we'll look at each of these individually now. So the testes or testicles, these are ovoid structures located outside the abdominal pelvic cavity, and a sac-like structure composed of skin, smooth muscle, and connective tissue, referred to as the scrotum. Each testes is divided into lobules or internal segments that contain tightly coiled loops, referred to as seminiferous tubules, and this is the site of sperm production. The testes perform 2 important functions: sperm production and the secretion of androgen hormones. And in this case, the primary androgen in humans is testosterone. The seminiferous tubules contain 2 cell types: spermatogenic cells, which are sperm-forming cells, and then substanticular cells, which support sperm production. You also have what they call interstitial cells or laid egg cells. These are between the seminiferous tubules, and they produce and secrete your androgens, such as testosterone, into the surrounding interstitial fluid around those particular seminiferous tubules. The myeloid cells, these are muscle-like cells that surround the seminiferous tubules. They contract to push the sperm and the testicular fluid through the tubules. Seminiferous tubules of each lobule will merge to form a single straight tubule that will move sperm from the ret test into the ret testes. Now, the ret testes is a network of tubules in the posterior testis. It transports sperm to the efferent ductules, which carry sperm from the ret testes to the epididymis. The epididymis is going to be the first portion of the duct system. So here we have our testes, and this is going to be the lobules right here, okay? The seminiferous tubules. And then here's our straight tubule, okay? And then we're going to go into the efferent ductules here of the ret testes. And then we have our epididymis right here. This is this segment here and then all along here. So the duct system is how sperm is going to travel. So sperm travels through a series of ducts bathed in semen secreted by accessory organs. This liquid will provide nourishment and support, and the ducts are involved in ejaculation, where sperm is expelled from the penis. So the epididymis is filled with ductules, and this is the site of sperm maturation and storage. On the superior and posterior surface of each testes, this includes the head, body, and tail regions. The head contains the efferent ductule of the testes and sits on the superior surface. Sperm moves from the head to the narrow body and into the small inferior tail. Sperm will then move from the efferent ductules into the single Ductus Epididymis that passes through the region of the epididymis, through all regions of the epididymis. The Ductus Epididymis is a long, coiled tube lined with pseudostratified epithelium cells and displays non-motile microvilli, or stereocilia. The microvilli will absorb excess testicular fluid, provide nutrients to the sperm, and complete the maturation process of the sperm. The length of the Ductus Epididymis allows sperm to be stored for months until ejaculated or reabsorbed. The Ductus Deferens, this begins at the tail of the epididymis where the Ductus Epididymis widens. This is also referred to as the vas deferens. The vas deferens, or Ductus deferens, is a long, thin tube that travels with the testicular arteries, veins, and nerves from the posterior border of the epididymis through the fibrous tunnel of the inguinal canal into the pelvic cavity, loops around the ureter, and passes over the lateral side and down the posterior surface of the bladder. And it will actually terminate in a sac-like structure called an ampulla. The mucosa of this hollow tube consists of pseudostratified epithelium and 3 layers of smooth muscle in the muscularis layer. During ejaculation, smooth muscle will contract, squeezing the sperm forward along the tract. The vas deferens can store sperm for months and reabsorb any sperm that has not been ejaculated over time. The ejaculatory duct, this is a fairly short duct. It's the next structure that receives the sperm from the vas deferens ampulla, location of the accessory organ. It's also the location of the accessory organ called the seminal vesicle. The duct will then also travel through the prostate on its way to the urethra. The urethra, we talked about this in the urinary system. This is a structure that is both reproductive and urinary in males, and it connects the urinary bladder to the external body surfaces. In males, it also transports both urine and semen through the 3 regions. We have the prostatic urethra, which surrounds the prostate gland, the membranous urethra, which passes through the external urethral sphincter, and then the spongy urethra, which extends through the penis and terminates at the external urethral orifice. So here we can see, here's the testes, here's the epididymis. Okay, here's the end of the epididymis, and this is the Ductus deferens, comes up, loops around the ureter, and then it will actually, this is the Ductus deferens ampulla, and then this will actually consist of the ejaculatory duct here, which will empty out into the urethra. And then you have the prostatic urethra, the membranous urethra, and then the penile or spongy urethra. So the penis delivers sperm into the female reproductive tract and has the following anatomical features. The external penis consists of a base called the root, the body, which is the shaft, which will terminate at the glans penis, where the external urethral orifice is located. Loose skin of the penis forms a circular fold called a prepus, or foreskin. This can be removed by a process referred to as circumcision. The internal penis is 3 cylindrical bodies of erectile tissue, the corpora, and this is covered by a dense fibrous connective tissue. Each erectile body a spongy network of connective tissue and smooth muscle that has lots of vascular spaces. The corpora cavernosa, this is to the ischial rami. And then the corpus spongiosum, this is a single piece that expands to form the bulb of the penis at the base. Sorry guys, got interrupted there, internet connection. So the corpus spongiosum, this is a single piece that expands to form the bulb of the penis at the base. It sits between the crura, where they form the root of the penis, and it will actually connect the penis to the pelvic bones. So the internal iliac arteries will provide branches, the internal penile arteries, which will supply blood to the penis. The internal penile arteries give rise to the dorsal and deep arteries. The dorsal arteries will supply the penile skin and fascia and corpus spongiosum, and the deep arteries will supply the corpus cavernosa. Sensory and motor fibers from both the sympathetic and parasympathetic nervous system will innervate the penis. Tactile pressure and temperature receptors are also abundant. Both are important for sexual stimulation and control of the smooth muscles that control the distance of the scrotum from the body wall. The penis and the scrotum will make up the external male genitalia, component of the perineum. Diamond-shaped region between the thighs, bordered by the pubic symphysis anteriorly, and the ischial tuberosities laterally, and the coccyx posteriorly. It's divided into the urogenital triangle, which contains the base of the penis and the scrotum, and the anal triangle, which contains the anus. There are various accessory sex glands. The seminal vesicles, these are paired exocrine glands on the posterior surface of the urinary bladder, near the ampulla of the Ductus Deferens. The duct of each seminal vesicle converges with the Ductus Deferens to form the ejaculatory duct. The exterior of each gland is enclosed by a fibrous capsule that covers a thick layer of smooth muscle. The deep mucosal layer is made up of pseudostratified columnar epithelium that will actually secrete a seminal fluid. Seminal fluid is a yellowish secretion that is going to make up 60 to 70% of semen volume. It is composed of fructose. This is a sugar that sperm utilizes for ATP synthesis. Prostaglandins, which will stimulate smooth muscle contractions in both genders, reproductive tracts, and increase sperm viability. Coagulating proteins and enzymes, both from prostate, which will combine to form a temporary clot of semen in female reproductive tracts. And the pH of the seminal fluid is alkaline to neutralize the acids in the male urethra and in the female reproductive tracts. Now the prostate gland, this is an egg-sized gland inferior to the urinary bladder, surrounds the urethra and the ejaculatory ducts. It's made up of 20 to 30 tubular glands dispersed among smooth muscle and connective tissue, and it's enclosed in the fibrous capsule. During ejaculation, the prostatic smooth muscle contracts. This will squeeze the prostatic secretions into the prostatic urethra and then out through several ducts. Now the prostate secretions, this will make up 20 to 30% of semen volume. It's a milky fluid that contains citrate, which is another sugar that sperm can utilize for ATP synthesis. PSA, which stands for prostate-specific antigen and other enzymes. This will dissolve semen clot into the female reproductive tract and allow sperm to proceed further into the tract, as well as antimicrobial chemicals, which will inhibit bacterial growth to decrease the risk of infection in reproductive female tract. The pH is alkaline to neutralize acids in the urethra in the female reproductive tract as well. The next gland is going to be the bulbourethral glands, or Cowper's glands. These are marble-sized paired glands at the base of the penis on either side of the membranous urethra. Each gland has a short duct that connects it to the urethra, will secrete a thick, alkaline mucus-like fluid that helps to neutralize acidic urine remaining in the urethra in response to sexual stimulation. These secretions will also help to lubricate the gland's penis during intercourse. So semen is somewhat sticky, whitish mixture of sperm, which makes up about 5% of the semen volume, and all fluids from the various anatomical structures sperm travel through. A typical ejaculate, which is the amount of semen expelled during one ejaculation, is between 2.5 and 5 milliliters in volume, and that volume will contain anywhere from 40 to 750 million sperm cells. 5 minutes post-ejaculation, semen coagulates due to the activities of the clotting proteins from the seminal vesicles and the enzymes from the prostate. This prevents semen from leaking out of the female reproductive tract. After 15 to 30 minutes, semen clot breaks up due to the inactivity of the PSA and other anticoagulates from the prostate. Sperm cells are gradually released from the clot, activated, and they begin their ascent up the female reproductive tract. The alkaline pH of semen helps to make sperm fully motile and to begin the process of capacitation. This is a process that enables sperm to penetrate and fertilize an immature female gamete. It is completed in the female reproductive tract where secretions prepare sperm cells, where the secretions prepare the sperm cells plasma membrane to fuse with that of the female gamete, the ovum. Prostaglandins will stimulate the contractions in the female reproductive tract and possibly thin mucus, allow the sperm access to further into the female reproductive tract, increasing the probability of fertilization. Antimicrobial chemicals from the prostate will also kill certain bacteria, including E. coli, which could be damaging to the sperm. The prostate slowly enlarges from birth to puberty, expands rapidly until about the age of 30. Further enlargement often occurs after the age of 45. If the prostate is non-cancerous but expands to the point of compressing the urethra, that condition is referred to as benign prostatic hyperplasia, or BPH. An enlarged prostate can be secondary to prostate cancer. Second most common cancer in the U.S. of men. An enlarged prostate can be secondary to prostate cancer. which again is the second most common cancer in U.S. men after non-melanoma skin cancer. It's one of the leading causes of cancer death among all men of all races. Risk factors for prostate cancer include genetic predisposition, African ethnic heritage, age over 50 years, obesity, and poor diet. Screening usually includes digital rectal examinations, as well as assessment of the prostate-specific antigen or PSA levels. Now, the thing with the PSA test is that you do oftentimes get a lot of false positives, in which case you need to follow up with testing. If there are any suspicious areas, such as lumps, or if the PS levels are above 4 nanograms per mil, a tissue biopsy specimen from different sites of the prostate are taken to look for any cancer cells. This may be treated with surgery, radiation therapy, chemotherapy, or hormonal therapy. In some men, tumors grow very slow and have low risk of metastasis. In these cases, regular monitoring may be all that's required. Some tumors are quite aggressive, however, and metastasize to other tissues rapidly. For this reason, screening and early detection are critically important. Okay. So now let's look at the scrotum. The scrotum is found between the thighs, and it contains and houses the testes. The midline septum divides the scrotum into 2 identical compartments, 1 per each testis. Raphe demarcates septum on the external scrota. This is a visible line that runs posteriorly to the anus and anterior along the vertebral surface of the penis. The scrotum wall contains an outer layer of skin, a superficial fascia, and a layer of smooth muscle. And the layer of muscle is referred to as a dartos muscle. The spermatic cord, this is a tube extending from the scrotum. It contains the vas deferens, blood, and lymphatic vessels and nerves, and that leads to the pelvic cavity. You also have the cremaster muscle, which is smooth muscle that controls the height of the testes. It enters the pelvic cavity via the external inguinal ring and terminates at the inguinal canal and leads to the abdominal cavity. Both the scrotum and the spermatic cord work together to support sperm production by regulating temperature. Normal cord temperature of 37 degrees Celsius is too warm for mass production of viable sperm cells. The scrotum is generally about 3 degrees cooler. The temperature regulation is managed by the following mechanisms. Blood vessels are arranged to and allow for heat transfer as arteries and veins carry blood in opposite directions. This reduces the scrotum temperature, an anatomical feature called the countercurrent heat exchange. The scrotum temperature can be increased or decreased by also altering the distance from the body. When cold, the scrotum is drawn closer to the body. When warm, the scrotum is relaxed and moves away from the body. The surface area of the scrotum is minimized in cold weather as well to decrease the heat loss. The surface area of the scrotum is increased when too warm, and this allows for greater heat loss and temperature decreases. So here we have all the structures of the male reproductive system and their functions summarized in this table. So let's talk about the physiology of the male reproductive system. Spermatogenesis is the process in which male gametes or sperm cells develop and is regulated by hormones from the hypothalamus and the anterior pituitary glands. Spermatogenesis begins at puberty and continues throughout life. It occurs in the seminiferous tubules by sperm forming or spermatogenic cells. The spermatogonia, these are the stem cells that initiate spermatogenesis. They have 46 chromosomes, they are diploid cells, and they're located in the basement membrane of the seminiferous tubules. Before puberty, spermatogonia divide only by mitosis to increase their numbers. After puberty, spermatogonia become fully functional stem cells that divide by mitosis, allowing one daughter cell to remain as a stem cell, while the other becomes a diploid primary spermatocyte. Following the events occur to each new daughter cell resulting from mitosis, the daughter that becomes the primary spermatocyte is pushed towards the lumen of the seminiferous tubule. The daughter that is to remain as a stem cell, the spermatogonium, replaces the original mother in the cell basement membrane. Each new primary spermatocyte will undergo first meiotic division, forming two smaller haploid cells that remain connected, and this is referred to as secondary spermatocytes. Both secondary spermatocytes will progress through the second meiotic division. Newly formed are four small round haploid spermatids that continue to move towards the lumen of the tubule. So here we have the process of spermatogenesis. In the seminiferous tubule, you undergo mitosis. One remains, one continues down, goes through meiosis 1, then meiosis 2, and we produce four daughter cells that are haploid. You also have what are called Sertoli cells, nurse cells, or sustenticular cells. They are going to surround the spermatogenic cells to provide support. The sustenticular cells are bound together by tight junctions. They extend from the basement membrane to the seminiferous tubule lumen. This microscopic anatomical arrangement forms the blood-testis barrier. This prevents the immune cells in the bloodstream from detecting newly formed antigens on the newly formed genetically unique sperm. Remember, as you go through meiosis, you are undergoing that crossing over process, and you are actually creating new unique cells that are not recognizable by the actual individual. They're not identical to that individual as well. The spermatogonia and the sustenticular cells are self cells that are able to interact with the immune system and other components in the bloodstream. These sustenticular cells will also provide structural support for stem cell development and secrete testicular fluid. This helps to transport sperm in the seminiferous tubules. It will provide nutrients for dividing and cells and produce androgen-binding protein and inhibin to help regulate spermatogenesis. They will phagocytize damaged spermatogenic cells and excess cytoplasm released from maturing spermatids. Spermatogenesis, this is the process of sperm maturation. This involves cellular shape and size changes. Begins in the testes, the spermatids elongate and shed their excess cytoplasm. An acrosome develops and covers the nucleus and helps with fertilization of the female gamete. The acrosome will actually contain digestive enzymes that will help it to penetrate that. Mitochondria increase in number and move to the developing flagellum site. Spermatids will separate from the sustenticular cells, and they are released into the seminiferous tubule lumen. The spermatids will develop a head, a midpiece, and a tail as they mature into mature sperm. The head will contain the nucleus and the acrosome and very little cytoplasm. The midpiece will contain lots of mitochondria that generate ATP, and then the tail is mostly your flagella. Sperm are still non-motile as they migrate to the epididymis, where they will complete maturation. This takes about 12 days to reach the epididymis tail, where the sperm cells will remain viable for months. The entire process, including both spermatogenesis and spermiogenesis, takes about 60 to 70 days to complete. Sperm cells that are not ejaculated within a few months will then be reabsorbed. And here you can see the progression from a spermatid to a fully functional sperm cell. Now, hormonal control of the male reproductive system is primarily that of testosterone. This is the main hormone that's involved in the regulation of spermatogenesis and male reproductive physiology. It's regulated by a multi-tiered negative feedback loop. It's going to start at the hypothalamus, then the anterior pituitary, and then the gonads. And this is what we refer to as the HPG axis, which regulates hormones involved in testosterone production and testicular function. Your first tier control, this is gonadotropin-releasing hormone, which is secreted by the hypothalamus. The second tier control is the anterior pituitary detects gonadotropin-releasing hormone and secretes FSH and LH. Your third tier control involves LH stimulation of the interstitial cells to produce testosterone and FSH stimulation of the subtesticular cells to secrete the androgen-binding protein and inhibin hormone. Ultimately, testosterone stimulates spermatogenesis, as well as the development of the male secondary sexual characteristics. Elevated testosterone and inhibin levels are sensed in the hypothalamus and the anterior pituitary, causing a negative feedback loop. And here's that HPG axis. Now, to reproduce, sperm must travel deep into the female reproductive tract, and sexual intercourse or copulation or coitus is a mechanism that optimizes chances of fertilization. An erection is an enlargement and stiffening of the penis that is caused by the engorgement of blood, and this will allow the penis to enter into the vaginal tract. In non-aroused state, the penis is flaccid or relaxed, as blood vessels supplying the penis are constricted. During sexual arousal, parasympathetic reflex triggers the release of nitric oxide from the blood vessels and endothelial cells. This will relax the arterial walls. The arterioles will dilate in the blood sinuses in the erectile tissue, and this will allow for a large volume of blood to enter into that tissue. Increased blood flow and widening blood sinuses will cause an erection. The erection is partially sustained by compression of veins that drain the penis. The corpus spongiosum does not become as rigid as the corpus cavernosa. This allows sperm and semen to travel through the urethra unimpeded. Ejaculation is the process in which we expel semen from the penis under sympathetic nervous system. So the erection is parasympathetic, ejaculation is sympathetic. And this occurs in two stages. You have emission, which is the movement of sperm, testicular fluid, and prostate and seminal secretions into the urethra. Expulsion occurs as semen, sperm, and fluids from other regions of the reproductive system accumulate in the urethra. This generates sensory impulses processed in the sacral portion of the spinal nerve. Motor neurons from the spinal cord stimulate rhythmic contractions of skeletal muscles at the base of the erectile columns of the penis, and this will actually elicit the expulsion of the semen from the urethra. Orgasm, this is a time period during which feelings of pleasure are experienced and coincides with ejaculation. Resolution or relaxation follows the orgasm and ejaculation. The blood vessels in the erectile tissue and blood sinuses constrict, forcing blood out of the penis. The refractory period is a latent period that occurs after ejaculation. It can vary in duration based on the individual, where a man can actually not achieve another orgasm. Male infertility, this is the inability to produce pregnancy after 1 year of unprotected intercourse. Approximately 40% of all infertility cases result from male infertility, usually due to low sperm counts. Normal levels of sperm are from 40 to 750 million sperm per ejaculate. Count of less than 15 million sperm per millimeter of semen is usually an indication of infertility. Low sperm count can actually result from any sort of damage to the testes, such as physical trauma, exposure to radiation or disease, or it could actually be due to developmental defects. During normal development, the testes begin forming inside the abdominal pelvic cavity and then descend into the scrotum. If the testes does not descend into the scrotum, sperm cells will not be produced. In addition, inadequate secretions of gonadotropin-releasing hormone, FSH or LH, or testosterone for any reason will lower sperm counts. Testosterone levels will increase dramatically at puberty, which will lead to the following changes. This usually begins around the age of 12 to 14. The hypothalamus becomes less sensitive to feedback inhibition caused by the low levels of testosterone and androgens from the adrenal cortex. The uninhibited hypothalamus will increase the secretion of gonadotropin-releasing hormone and stimulate the activation of the HPG axis. Increased testosterone levels will trigger spermatogenesis and the appearance of the secondary sexual characteristics. Once spermatogenesis begins, males are capable of sexual reproduction. Your secondary sexual characteristics first develop primarily at puberty and will continue through early adulthood, and include growth of the pubic, axillary, and facial hair, as well as increased hair growth in specific regions of the body, such as the chest. The larynx enlarges and the vocal cords thicken. This results in a noticeable Adam's apple and deepening of the voice. The skin thickens and the sebaceous glands will increase in secretions. Testosterone causes the somatic effects, of many of which are anabolic in nature. Bone density increases, muscle density increases, erythrocyte production increases as testosterone increases erythropoietin secretion. Testosterone influences over behavior. It's the basis for male libido or sexual drive. Effects of aging on the male reproductive system. Male climacteric or andropause. This is a period in which reproductive function begins to decline in men. Changes typically occur gradually, sometime in the 5th decade of life, but vary widely amongst individuals. The size and weight of the testes may decrease. The number of testicular cells and interstitial cells may also decline. The subtesticular cells will secrete less inhibin, and interstitial cells will produce less testosterone. Both lead to an increase in gonadotropin-releasing hormone as a negative feedback mechanism is not triggered, and this will lead to the production of SSH and LH if these levels remain elevated. Indicates the testes are not producing enough testosterone, which can be treated by providing exogenous testosterone. So reduced levels of testosterone are associated with a gradual decrease in the production of sperm cells, depressed mood and fatigue in some individuals, loss of muscle mass, and decrease in bone density. The prostate gland will result in hypertrophy. Is one of the most notable changes during this process. Often compresses the urethra, leading to difficult urination. Erectile dysfunction, or ED, can result from physical or psychological stressors. Psychological influences such as stress, depression, and anxiety can lead to ED. Physical stressors could be cardiovascular disease, diabetes, obesity, use of tobacco or alcohol, and certain prescribed medications can also play a role. Older men have a greater risk because they are more likely to have related physical conditions. Also, the amount of connective tissue and erectile tissue of the penis increases with age, reducing blood flow to the penis. Treatments include medications that permit blood vessels supplying the penis to remain dilated, therefore maintaining an erection. Also, surgery may be used to repair damaged blood vessels and insert penile implants. And that will be the end of this recording.